our G protein has already been activated and separated. So we have our beta and gamma subunit and our alpha subunit, the GTP. So this is our common set. This is what we're going to do with every aspect. Remember, we're in a lipid membrane. So all of these are just moving around randomly. So when they come in contact with, the receptor comes in contact with the hormone, or the alpha subunit comes in contact with the membrane-bound enzyme, that's still random. Okay? There's no attraction, positive or negative charges. It's just moving around the membrane and happens to bump into this molecule. And it also is a membrane-bound molecule. And then this first sequence, this is an enzyme known as adenylate, let me write that up a little higher, adenylate cyclase. You may also see adenylyl. They're both the same. Over 90% of the time, enzymes are going to be identified with ASE. So if you see the ASE, it is an enzyme, right? Not all enzymes will have ASE, but if it has ASE in its name, it definitely is an enzyme. The alpha subunit, okay, is also an enzyme. This G protein is also an enzyme, even though it doesn't have ASE in its name. It's taking GTP and breaking it down to GDP once it contacts this and provides the energy. What molecule did we talk about, or did you learn about last semester, that is an ATPase? This is G protein, it's a GTPase. What molecule did we talk about that is an ATPase? It doesn't have ASE in its name. Think muscle activity. We talked about muscle contraction. What molecule breaks down ATP to ADP? Myosin. Remember the myosin head? Binds to ATP, and ATP is broken to ADP and releasing the energy for changing cocking the head. All right, so in a similar fashion, the G protein is doing that to GTP. So even though we don't talk about it as an enzyme, that's the way it's functioning. All right. So adenylate cyclase is an enzyme. It's membrane bound. There is the active alpha subunit, and GTP is now going to connect with that, where it would not have been able to do that as a complete GTP protein. So when our G protein is bound to the receptor as a whole 3 subunit, interaction with adenylate cyclase doesn't cause any change. So now the adenylate cyclase with the GTP is going to be modified. GTP is going to release phosphate, and energy is going to be utilized to activate this enzyme. Now it's a functional enzyme. By itself, as adenylate cyclase in the membrane, it was inactive. But now it's changed its shape, and it is going to be able to function by taking a molecule of ATP and using the enzyme kinetics, to convert this to cyclic AMP. In other words, 2 phosphate molecules are removed and it's converted to a ring formation. And so this is often abbreviated with just a C, cyclic AMP. So this is a second messenger. The alpha alpha subunit is not a second messenger. Too low. The alpha unit is not a second messenger. Does that help? The adenylate cyclase is not a second messenger, but cyclic AMP can now move throughout the cell and interact with other enzymes and other molecules. That's why it's given the name second messenger.
So cyclic AMP is the second.
Yes. Okay. So this is where we are on the diagram. Cyclic AMP. And now cyclic AMP functions to activate hundreds of other enzymes in the cell known as protein kinases. And in this case, it's a specific protein kinase A. So cyclic AMP will interact with protein kinase A, change its shape conformationally, so chemically it can activate other molecules. And what this does is it adds phosphate groups. Phosphorylation. So when we take ADP and go to ATP, that's phosphorylation. So there are many enzymes in the cell that are either turned on or turned off by the addition of phosphate groups. Okay. So this will phosphorylate cellular enzymes. It interacts with it. It may not stay bound to it completely, but it interacts with it enough to activate it. And then now the protein kinase is an active enzyme. Just like alpha GTP interacts with this, it doesn't stay bound to it, it will then come off. And as GTP, it will reverse, go back to its G protein, and then that's ready to be used again. So the alpha subunit and its GTP, once it binds and interacts with adenylate cyclase, won't stay there. But when it leaves this as an active enzyme that will then take ATP to cyclic AMP then activates protein kinases, and the protein kinases phosphorylate. We have a cascade. And remember we talked about the ionotropic and the metabotropic receptors? Ionotropic are fast acting, but they're limited to that one membrane spot. Okay? Whereas this may take a while to get things going in the cell, but it's amplified. Multiple cyclic AMPs can be formed by a single adenylate cyclase enzyme. And multiple protein kinases can be activated by a single cyclic AMP. So it takes a little bit longer, but we have this amplification of the effect. So this is where we are at right here. Okay, it's a phosphorylation of the enzyme. Obviously, we don't want this going on forever and ever. And so there's multiple places to interfere with its activity. The hormone can separate from the receptor. And that's going to take a while for half-life to decrease the hormone. So as that hormone separates, another hormone will take its place until over hours, the number of hormones in the blood decrease. Okay? We can remove the G protein. We can remove ATP. But there's another enzyme right here that works specifically on breaking down the cyclic AMP and returning it to ATP status. So phosphodiesterase functions to degrade cyclic AMP. So if you're making flashcards for these individual molecules, right, you would want to include what they interact with. So if you have a flashcard for the alpha subunit that interacts with adenylate cyclase, You would want to include whether they're membrane bound or in the cytoplasm. All right. Phosphodiesterase is not membrane bound, it's in the cytoplasm. Cyclic AMP, protein kinase A are in the cytoplasm. Our G protein and our adenylate cyclase are membrane bound. There are advantages to moving to another room. So this is norepinephrine.
And therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
So the phosphorylase in the liver cell may be different enzymes in other cells. Okay? Yeah.